Hello, this is Jason Rowe. Today, I'm going to be talking about water, urea reabsorption at the kidney, how our body recycles water, our internal water, and filters out waste products, keeping our system running incredibly effectively. Um, so, how it does this in the kidneys with aquaporins, abbreviated AQP, called AQPSs. These are a family of membrane-spanning proteins that facilitate the transport of water across cell membranes. They are found in all tissues of the body, but they are particularly abundant in the kidney. The importance of these channels in the kidney is very easey you can quickly recognize because the absorption of water and the concentration of glomerular filtrates is an essential function required to sustain life in mammals. For example, in humans, our kidneys will filter through about 150 liters of aqueous fluids, our body fluids, each day, and only around 2 liters of that material is voided out of our system as urea. So this shows how important these functions are to just our existence. The kidneys are responsible for filtering blood and excreting waste products from the body. So in order to do this, the kidney has to be able to concentrate urine. AQPs play a critical role in this process. They allow water to move from the filtrate, the body fluids going into the kidneys. They allow it to move to the collection ducts where it can be excreted as urine. Here in table 1, we have a showing of the location and function of the various aquaporins in the kidneys. There are 7 AQPs that have been identified in the kidney. Each aquaporin has a distinct location and function. AQP1 is found in the apical and basolateral membranes of the proximal tubule and the descending thin limb of Henle. It is responsible for reabsorption of water from the filtrate. AQP2 is found in the apical membrane of the principal cells of the collection duct. This one is responsible for the final concentration of urine before it is accessed from the body. AQP3, this one is found in the basolateral membrane of the principal cells of the collection duct. This one helps regulate the flow of water between blood and the interstitium. AQP4 is found in the basolateral membrane of the distal convoluted tubule and in the collection ducts. It helps regulate the flow of water between the blood and the interstitium. AQP6, this one's found in the apical membrane of the thick ascending limb of Henle. It helps to regulate the reabsorption of sodium from the filtrate. AQP7, this is found in the apical membrane of the intercalated cells of the collecting duct. It helps transport water and glycerol. And last but not least, AQP11. This is found in the apical membrane of the segment of the proximal tubule. This helps regulate the pH of the urine. Like all of our body, we need a regulated, monitored pH that stays stable within the body. So while all this water exchange is going on, we don't want the pH to get out of sync with the rest of the body, which could cause issues. Here we have a very complex example of the regulation of water retention by vasopressin. Vasopressin is a hormone that's released from the pituitary gland in response to the body sensing dehydration. So vasopressin binds to the receptors on the principal cell of the collection duct, and it triggers the insertion of AQP2 into the apical membrane. And when this happens, this allows water to be reabsorbed from the filtrate, and that helps to removing that water helps to concentrate the urine before excretion. So in this figure, these blue ovals that you see up here, all those blue ovals are AQP2. And then the interaction of vasopressin with either the cells of the receptors or the vasopressin receptors in the basolateral membrane of the principal cell results in the incorporation of AQP2 into the apical membrane. So that's how it gets brought into the area where it needs to be to help with that water reabsorption. Basic workings. There can be modifications after the proteins have been created. AQPs, especially AQP2, undergo different modifications after they've been synthesized, which impacts their function. These modifications include glycosylation, phosphorylation, and ubiquitination. Pardon me. These changes affect where the AQPs are located within the cell, how they function, and how they are processed. One specific modification in phosphorylation of a segment of the gene for AQP2. AQP2 plays a role in promoting movement from vessels inside the cell to the plasma membrane. The effects of this phosphorylation on water transport regulation and other aspects is still being debated. I'm not quite sure exactly how it works, but we are learning. Most of the AQPs found in the kidneys have specific sites where sugar molecules can attach. AQP 1, 2, 3, and 4 can partially or fully be glycosylated. The exact role of glycosylation in these AQPs is uncertain. In AQP2, glycosylation may be important for certain cellular movements, but blocking the glycosylation doesn't affect the protein's functionality. Ubiquitination of AQPs leads to their internalization and subsequent degradation or breakdown. A small molecule called ubiquitin can attach to a group of APQ2 proteins, and it's proposed that when this happens, the AQP2 is sorted into vesicles within the cell, these multivesicular bodies, and then targeted to lysomes for degradation, breakdown, or to exosomes for external release. Also, mutations in the AQP2 genes can lead to a variety of kidney diseases. So, for example, if there's a mutation in the AQP2 protein, this can cause nephrogenic diabetes insipidus, a condition in which kidneys are unable to concentrate urine properly, leading to excess urination and thirst. So if the body can't properly remove water from the waste efficiently just to create urine and get out of the body, if it's not doing that efficiently, we're going to not lose all the waste materials that we need to lose, and we're going to lose excess water, causing you to urinate frequently and be thirsty. Mutations in the AQP1 can also cause kidney problems. An example, there's a mutation in the AQP1 that is linked to the risk of kidney stones. Your body is more likely to produce kidney stones, which are can be very painful to deal with. So changes in the AQP function can lead to activity with diseases. A variety of diseases can affect the function of AQPs. Here in table 2, we see a plethora of them. Kidney diseases such as glomerulonephritis and nephrosclerosis can lead to a decrease in AQP2 expression. This can impair a kidney's ability to concentrate urine. So it's not, in this case, it's not that the aquaporin is not being released properly. It's that there's a mutation in the protein itself, and because it's not shaped right, so to speak, the kidney is not able to use it properly, and therefore it's going to have problems concentrating the urine in the case of AQP2. Diabetes mellitus can also affect the function of AQPs. Like diabetes can lead to an increase in AQP1 expression in the glomerulus. This can increase the risk of kidney damage as well. So we need all these proteins functioning properly in order to do their job. Wrapping this up, there are plenty of future studies in this area. Research on AQPs is ongoing. There's a lot of material out there. Future research is likely to focus on areas like identifying new types of aquaporins and their function, or new functions of existing AQPs. Development of drugs that target AQPs for treatment and kidney disease, whether those proteins have been mutated or aren't functioning properly. Different drugs can target those different areas to help our body, our kidneys, work properly or better. And then the use of AQPs as biomarkers for kidney disease, marking certain ones, especially if they have a possibility of a mutation, to see where exactly an issue may be coming from somebody who has kidney disease, to be able to narrow it down. So to sum everything up, AQPs play a critical role in the kidney's ability to concentrate urine. Because of these proteins, the kidney functions properly to filter our water efficiently into our system and remove waste products in the form of urine that we can excrete from our bodies. Mutations in the AQP genes and changes in the AQP function can lead to a variety of kidney disease. Future research on AQPs is likely to lead development of new treatments for kidney disease. It's an interesting and growing area. Thank you so much for taking time to watch.